my next experiment is for indicator. Now I have made one natural indicator using turmeric powder. So it is nothing but a turmeric solution. Using the turmeric solution, we are going to see that the acidic and basic nature of any substance. So in presence of acid, what is the change in color, and in presence of base, what is the change in color of the turmeric solution. It is a yellow turmeric solution. I am taking the turmeric solution in one test tube. Now I am adding, it is dilute H2SO4, better we know it is an acid. I am adding that acid slowly, just a few drops. Observe, there is no change in color. That means yellow turmeric solution remain yellow. So there is no change in color. But in presence of base, in a next test tube, I am taking the turmeric solution again. As we know, the turmeric solution is yellow in color. Now we are going to observe, is there any color change in presence of base or not. So with me now it is sodium hydroxide. It is a base, strong base in laboratory. Now I am adding a few drops of sodium hydroxide and see the change in color. It is deep red or reddish brown in color. So in presence of acid, it is actually yellow turmeric solution remain as it is, that means yellow, but in presence of base, it changes its color to reddish brown. So by that we can observe the acidic and basic nature of any substance. Now I have one synthetic indicator also. It is the phenolphthalein solution, and we are going to observe what is the change of color in presence of acid and base. So in one test tube, I am taking that acid again, that means the dilute H2SO4, and I am adding 1 to 2 drops of phenolphthalein indicator, just 1 to 2 drops, it is sufficient. Ultimately, actually there is no color change. There is no color change, though it looks actually hazy, but it is actually transparent solution, no color change is there. But in presence of base, I am taking base in another test tube, that means our same sodium hydroxide solution in another test tube. I am adding just 2 to 3 drops of phenolphthalein indicator. There is a deep pink color. So with acid, it is actually colorless, and with base, it is actually deep pink in color. By that way, we can see or we can observe the acidic and basic nature of any substance. This is a deflagrating spoon. Using that deflagrating spoon, I am taking sulfur powder, and we must burn the sulfur powder. Now sulfur bonds. I am taking that sulfur inside a glass jar, keeping that vapor inside the glass jar, and the gas is nothing but sulfur dioxide gas. Now with a small water, dissolve the gas. And we are going to test the acidic or basic nature of that solution. I have blue litmus paper with me, and when I dip blue litmus on that solution, the blue litmus changes into red. So when we burn the sulfur powder using the deflagrating spoon, it will form the sulfur dioxide gas first. When we dissolve the gas in water, it will produce sulfurous acid. So in presence of acid, we know the change of color of indicator paper, or that our litmus paper, blue litmus changed into red.